All right, this week we're moving on to a different type of cancer with a similar treatment to the one that we talked about last week. And this week it's going to be glioblastoma. So, as a little bit of background on this, I wanted to go over some basic, neural cell biology to lay the framework for all of it. And the name glioblastoma comes from glio from glial cells. And the concept here is in the brain and the nervous system in general, there are two major categories of cell types. The first is neurons, which get most of the attention because neurons are the cells that are actually sending signals in the nervous system, computing information, taking in sensory stimuli, activating muscles. Like most of the interesting, exciting action that happens in the nervous system is because of electrical signals that are sent via those neurons. The second major category are called glial cells. And all of these cell types, microglia, astrocytes, ependymal cells, oligodendrocytes, those are in the CNS, the central nervous system, meaning the brain and spinal cord. And there are a few other types out in the peripheral nervous system as well. And they have a wide variety of different cell functions. The thing they have in common is they're often considered supporting cells, meaning they don't actually send signals themselves, they don't compute any information, but they're absolutely critical for brain function, right? Oligodendrocytes surround axons and myelin, which is an insulating substance that acts, almost like rubber or plastic does around an electrical wire. Astrocytes control the metabolic environment and also form the blood-brain barrier. Ependymal cells produce cerebrospinal fluid. Microglia are effectively the immune cells of the brain, acting much like macrophages do. And so, they're not neurons. They're maybe not as exciting from a neurology standpoint, but still incredibly important. And they're also the source of most brain cancers because, unlike neurons, glial cells actually divide. Right? Neurons are generally very stable. They don't go through cell division. And so they're usually very low risk for mutations because most mutations happen during the cell cycle. All right. So there are a few bits of information here that I think are important. The first is, in the long run, all cells have to be derived from stem cells. It's just that most of them, like neurons, are derived from these neural stem cells and progenitor cells during embryonic and fetal development and childhood. And so this route, the vast majority of neurons in your body have already formed by the time someone is an adult. And there are a couple exceptions, a few small regions of the brain where neurons really do divide and get replenished from these neural progenitors. But for the most part, this is stable, it's stopped. That's why neurons generally don't derive tumors. Right. On the other side, these glial progenitor cells and stem cells, they are making new glial cells, especially astrocytes and oligodendrocytes. And one of the leading hypotheses about how, cancer in the brain really starts is it's usually derived from those stem cells, which is kind of ironic because stem cells hold the power to treat a lot of diseases, but unfortunately their ability to divide also makes them more susceptible to mutations and cancer. And so the idea here, this clonal evolution model is the idea that you start off with a mutation that makes a cell somewhat cancerous, and that begins your tumor. But then in the division process, you eventually get mutations that start giving you different varieties of tumors, and those mutations keep sort of branching off, almost like a malignant family tree, where you get this situation in the end that gives you not just one homogeneous tumor, but many different cell types within the tumor. That makes them very difficult to treat because now you've got multiple mutations that would be either resistant or susceptible to a variety of different treatments. And even further than that, like a true evolution model of cancer means that you have these populations of cells that intermix with each other. And so you end up getting these very, very diverse tumors, and it's almost impossible to find one particular therapy that's going to be effective for all of them. Okay. And layered on top of that, a mass of glioblastoma, unfortunately, is not just glial cells. So you will have, by the time the tumor grows to a substantial volume, you're going to have a lot of cells involved. You're going to have the actual tumor cells themselves, of course, the original stem cells that were not all mutated, a variety of different immune cells, macrophages and T cells that are recruited to the site because of the inflammation and potentially, neoantigen production that comes along with all of these mutations. Right, combine that with the growth of new blood vessels. Remember, endothelial cells are just the cells that line a blood vessel. And so we've got all of these potential cell types that we can target. We can target the tumor cells themselves. We can try to encourage these immune cells to attack more aggressively. You can also try to block the division of these endothelial cells to prevent blood vessel growth, because for a lot of tumors, access to blood flow is one of the limiting factors that governs how fast they can divide. All right. So in this case, we're going to be looking at a direct target of the tumor cells themselves, which is very similar conceptually to the antibody-drug conjugate and the EGFR target that we saw recently. And so we're still on that same target. It's a different antibody. So remember, whenever the drug ends in MAB, that's our monoclonal antibody, and that antibody binds to EGFR. And a little bit of notation here. EGFR is being shown here like this is full EGFR, and that is also full EGFR. This EGFR mutation, that is a mutation where several exons from the EGFR mutation or gene are removed, and it makes a shorter protein, but unfortunately it's not non-functional, it's overactive. And so that's going to be a big part of this. Is there are a few different EGFR mutations that can drive cancer growth in glioblastoma. This one is going to be our major focus because it makes a constitutively active receptor. And that just means that the receptor is always turned on, even if there's no EGF present, right? Even if there's no ligand, receptor is always on. That stimulates the growth pathway. And so, right, we're using an ADC, right, our antibody linked to a drug that, exactly like the previous ADCs that we saw, our drug target or our payload is targeting microtubule formation, which breaks apart this mitotic spindle. Cells can't divide, and if that becomes significant enough, cells will go through apoptosis and die off. Okay, so I'm going to go through at least parts of two different papers. This first one is more of a basic cell biology and in vivo mouse study where they're trying to characterize the effects of our ADC on glioblastoma cells in culture and in mice. And so the question is, of course, does this drug kill glioblastoma cells? And if it only kills a subset of those cells, what's the differentiator that determines which cell types go down? And so what ended up happening is, our drug ABT 414 is the ADC, the combination of our monoclonal antibody and our payload, does kill glioblastoma cells, but only if there's EGFR overexpression of some kind. If cells just have normal, typical levels of EGFR, then the ADC isn't effective because clearly that's not the mechanism that's really driving the growth of those cancer cells. Right? And then this EGFR Y1068, Y is the single letter abbreviation for tyrosine amino acid at position 1068 in the protein's amino acid chain. And so the cell has to overexpress EGFR for our drug to work. It does not necessarily have to have this phosphorylation marker, which is effectively just another marker of overactivity. All right, so our goal here, test this out on glioblastoma cell lines. And, right, so these terms, MAB 806, that is the rather boring name for the actual antibody itself. This monomethyl auristatin F is the payload that's inhibiting microtubule mitotic spindle formation. And So we're looking at glioma cell lines that have a variety of different conditions. So some of them overexpress wild-type EGFR, meaning they just make a lot of the normal wild-type receptor, or they have that EGFR V3 mutation. That's the one where 3 different exons have been removed and leads to a constitutively active receptor that's always on. Okay, so data. LN229 is the glioblastoma cell line that was used in this case. And the control just means they haven't done anything to increase EGFR expression. There are no mutations. It's not constitutively active. And they're comparing PBS, which is effectively the negative control. AB095 is effectively a control antibody that they don't expect to really have any effect, but it's just really being extra certain that it's not some random effect of an antibody, that it has to be our antibody. And then, of course, ABT414 is our actual experimental condition that we're hoping to have an effect. In these control cells that don't specifically overexpress EGFR in any way, there is no statistically significant difference between any of the conditions, meaning without the EGFR overexpression, nothing meaningful happens. All right. If you have EGFR overexpression in those cells, there is still really not that dramatic of a difference. You see some extension early on, but eventually they pretty much even out with each other. When the EGFR V3 mutation is added, right, and it's constitutively active now, so not only do you have a lot of the receptor, but you have mutant receptors that are always turned on, then you start seeing a very dramatic difference where these cells that got the negative control treatment, lasted all of 40 days, whereas the ones that had the EGFR mutation lasted about 100 days. Right. And just for clarification, the survival, that's not survival of cells, that is survival of mice, right? Because this was effectively a glioma model where they took these glioma cell lines and injected them into the brains of mice. And then they were doing a survival study on how long the mice lasted after being exposed to those cells and either the negative control or the treatment. And so the conclusion for right now appears to be, this drug can be very effective if patients have this constitutively activating EGFR V3 mutation. If they don't, and EGFR is not the driver, then unsurprisingly, right, there's no meaningful effect. Okay. All right, so let's try this out in humans. Okay, so this is, right, same drug, that's our ADC, and we're testing it out in a phase 3 randomized trial in, right, EGFR amplified newly diagnosed glioblastoma. So they're looking for patients that got a recent diagnosis, and that's critical because unfortunately glioblastoma does not have a long survival rate at all. So it's important to move quickly. But it's not a follow-up, it's not a second treatment. First round treatment for EGFR amplified, meaning patients with tumors that have an EGFR mutation as the driver of their glioblastoma. And the sample size here was impressive. N of over 2,000 individuals. Unfortunately, a decent number of them were excluded because variety of reasons. They were screened for EGFR amplification, and it just wasn't present. The patient backed out and just didn't want to. Follow-up didn't confirm the type of tumor that they thought they had. You get the idea. So lots of reasons to filter people out. But since they started with a relatively large number at the beginning, right, you still wind up with a fairly sizable N number of 639, right? And they were randomized into control versus placebo, placebo versus experimental. Obviously, the placebo not receiving treatment, and the other half randomized to receive our ADC. And then, we're measuring disease progression, survival rates, toxicity, right? Some sort of measurable side effect, which can be difficult to determine because it's hard to know what's because of the cancer and what's because of the drug. And then, of course, some withdrew probably because they got too sick and didn't want to tolerate it anymore, or progression just worsened pretty badly. Okay. Unfortunately, and somewhat discouragingly, when you look at all patients combined, these curves are not meaningfully different from each other. Notice the p-value of 0.63 is basically shows nothing meaningful happened. It's impossible to detect a difference between the control and the treatment group. And that was for overall survival. This was for what they called progression-free survival, meaning survival and not having progression or worsening of the tumor. And there was a significant difference here, right? 0.03 is considered a significant p-value because there was some separation of that curve in the middle. But this is a great illustration of, do you have statistical significance? Yeah, 0.03 is statistically significant because there was a decent sample size. Do you have clinical significance? This is a pretty small change in survival rate. If we compare these, right, you're talking about going from, I don't know, maybe in the low 30s down to 25%. And so, yeah, a decent number of patients got maybe a couple months of life extension out of this, but it really wasn't that dramatic. Okay, so the interesting follow-up question from that is, when we look at these patients, some of them may have responded, some of them didn't. What if we look a bit more closely at exactly which subcategories of patients may have done better than others, because that could give you more interesting trends that you don't see when you just lump everyone together like this. All right, and it turns out the answer to that question is sort of. So we've got a couple things going on here. We're looking at, all right, overall versus progression-free survival. So A and B are both overall survival. B and D are both progression-free. And then we're looking at patients that either do or do not have the EGFR V3 mutation, right? So we're subdividing those patients, and A and B are with the mutation, C and D are without. And so if you go through and look at the p-values, we get not much that's interesting, and any of these three p-values are all relatively high, meaning there's no discernible difference between the two groups, right? And remember, red lines are with drug, blue lines are placebo. In B, right, this is progression-free survival with people that do have the EGFR mutation. And, 0.002, again, it's a statistically significant p-value. And so the clinical question is, all right, if you pick the point in here where there's the biggest difference, the difference between this line and this line is running at a rate of, okay, if that's around 40 and this is around 25, about 15% of people right above the placebo rate are alive during this range. The unfortunate thing is it's a fairly small time window. So we're really seeing our benefit in here right in the 6 to 18 month range. And unfortunately, by the time we get out to 24 to 30, there's no real meaningful difference anymore. And so this is a case where the drug is extending life expectancy for, it appears about 15% or so of these patients that have the EGFR V3 mutation with progression-free survival, but it's not really changing overall survival rates all that significantly, and it's only during that fairly short window within about an 18-month range, whereas once you get out past that, there's no difference anymore. And so I think this really highlights one of the most difficult aspects of oncology and cancer treatment is deciding, okay, how big of an impact do we need to make before it's worth the time and the effort and the money. And unfortunately, my conclusion is I think if you showed these data to most patients, it probably wouldn't be enough to convince them that going through all of this is worth it. But that's kind of an individualized opinion and not really a yes or no, like black and white fact kind of question. Okay. So to put that in the broader context, I think what's happening here is glioblastoma is known for being among the most difficult cancers to treat. You can see, I mean, these people were like, this is not a follow-up. This isn't them receiving a second treatment. This is first line. And, you know, from day zero of being treated, which is probably very soon after diagnosis, very, very few of them are left by the time we reach the end of the study. And so, you know, is this a dramatic, you know, wow, I can't believe this happened result? No, in one sense, but yes, in another, in that treatments for glioblastoma have been so non-existent that oncologists and physicians are viewing this, I think, as it's big because it's the first time that anyone has really moved the needle at all. And so this definitely isn't the end goal and nobody's happy with it. But it at least shows a proof of concept that these drugs, maybe if they're improved and developed a little bit further, have the potential to, you know, further down the road, possibly make a bigger impact.